started with a new chapter which is in 12th NCERT, second chapter, sexual reproduction in flowering plants. In the previous lecture, we discussed introduction to the chapter, and then we were talking about the male reproductive organ. Male reproductive organ is stamen. What does the stamen contain? Stamen contains two parts, right? So that we will continue in this lecture also. So we are talking about 2.2.1 in NCERT. It talks about stamen. What does the stamen contains inside is microsporangium, and microsporangium, what does it releases means pollen grains. So this is the side heading, pollen grains. Now this we will discuss in today's session. Now we already discussed in the previous lecture that stamen is a male reproductive organ. Stamen is a male reproductive organ and stamen contains two parts, right? So what are the two parts of the stamen? It has a long slender stalk which is called as filament, and it has a lobed structure, a broader knob-like structure which is called as anther. Now we are talking about the anther in the previous lecture. Now where is anther located? Anther is attached to the distal end of the filament. How is the anther? It is broader, it is knob-like, it is the fertile part. Why are we calling anther as a fertile part? Because it is producing microspores. So anther, it is broader, knob-like, fertile part, right, of stamen. Usually, how many anther lobes will be there? Means generally the anther is two lobed and both the lobes are connected by connective. Generally, anther has two lobes which are connected by, both the lobes are connected by a sterile tissue. Since you are telling anther is a fertile tissue, the two anther lobes are connected by a sterile tissue called connective. What is the sterile tissue called? The sterile tissue which is connecting both the anther lobes is called connective. Now this connective tissue is supplied with vascular strands. So connective is supplied with what? It is having vascular tissue. It is having xylem and phloem, mean to say. It is connected with, it is having vascular tissue. When we draw the picture of microsporangium and anther, you can understand where is connective, where it is located. And we are telling that anther is a knob-like structure. Generally, a stamen has two anthers. So means it is bilobed. How, when can we see the bilobed nature of the anther? Means the bilobed nature is very distinct in the transverse section, TS, right? So we can tell that the bilobed nature of anther is very distinct, is very much clear, is very distinct when in the transverse section of anther. So when we make the transverse section of anther, the bilobed nature is very much distinct. That we will see it on the board, we will draw the diagram. And when we see the bilobed nature, when we see the transverse section, we can tell that anther is four-sided, right? So if we draw the picture, so let us make the diagram of the transverse section of the ovary. This is one lobe and the second lobe. Both the lobes are connected by a sterile tissue called connective. This is the sterile tissue connective filled with parenchymatous cells. So this we can call it as connective, which is connecting both the lobes of the anther. Now, is the anther having four sides? 1, 2, 3, 4. That's what we are telling. Anther is tetragonal. What do you mean by tetragonal? Four-sided. Now, it is having 4 microsporangias situated at the corners. So, if we have to see the microsporangia, this is one microsporangia, second microsporangia, third microsporangia and fourth microsporangia. So, it is having 4 microsporangia located at 4 corners. The anther is having 4 microsporangia. It is having 4 microsporangia located at the 4 corners. And this much is the anther wall. From here to here is an anther wall. The anther wall is made up of 4 types of cells. The outermost layer is called as epidermis. The outermost layer of the anther wall is called as epidermis. We are telling it's made up of 1, 2, 3, 4 layers. The outermost layer, I am telling it is epidermis. The second layer is called as endothelium. The third layer will be called as middle layers. The fourth one is called as tapetum. So, from here to here is an anther wall. The anther wall is made up of 4 layers. The outermost layer is called as epidermis. Inner to the epidermis is endothelium. Inner to endothelium are the middle layers, and the innermost layer, which is enclosing the sporogenous tissue, is called as tapetum. Quickly let us draw all the layers, right? So, if we have to show the epidermis, we told epidermis is single layered barrel shaped cells, right? So, this is epidermis. Single layered epidermis. Children, what is the role of the epidermis? Epidermis helps in protection, right? The function of epidermis will be always protection. This is epidermis, single layered, tightly packed cells. Now, function we are telling it is protection. Coming to endothelium. So, endothelium consists of hypodermal cells. It is made up of hygroscopic cells, we can tell. The endothelium is made up of hygroscopic cells. What do you mean by hygroscopic cells? They can hold water, they can lose water. So, they react to the water. So, that is why they are called hygroscopic cells. And these hygroscopic cells, how are they arranged? They are radially arranged, and in between the cells are connected by callose walls. They have callose walls also. Now, if we have to draw this endothelium, it will be below the epidermis. So, we can draw it like this. So, little thicker. We will make the endothelium little thicker. We are telling these cells are hygroscopic cells. They can retain water and they can lose water. They are the main cells which help in dehiscence. These are the main cells which help in dehiscence. Dehiscence means splitting of the anther walls. See? So, these cells are called as, what are this layer of cells called as? This is called as endothelium, made up of hygroscopic cells. What do you mean by hygroscopic cells? Hygroscopic cells means they can hold water, they can dehydrate also, they can lose water, and endothelium plays very important role in dehiscence. Endothelium plays, we can write here, important role in dehiscence. What do you mean by dehiscence? Splitting of the anther walls to release the pollen grains out. So, first layer, children, epidermis, protection. Second layer, endothelium, it helps in dehiscence. They are hygroscopic. So, they have thick walls, callose layers they have. Now, coming to the middle layers we are telling. This means 1 to 5 layer cells will be there. Made up of loosely packed parenchymatous cells. The middle layer, it is made up of loosely packed parenchymatous cells. It is made up of loosely packed parenchymatous cells. Now, let us draw the loosely packed parenchymatous cells. So, 1 to 5 layers we told. So, we will see how many layers we can draw here. So, this is one layer, right? Another layer also we can. They are loosely packed parenchymatous cells. Now, they also function along with the endothelium. If endothelium is dehydrating, these loosely packed parenchymatous cells also will dehydrate, means they also help in dehiscence. So, I can draw one more layer, I think. Yes. 1, 2, 5 layers of loosely packed parenchymatous cells. What do they make? They make middle layers, right? So, this is the middle layer. Now, the last layer, the innermost layer is called tapetum. Now, this tapetum consists of cells which are thick, which are big, which have prominent nucleus, which have more than one nucleus. The tapetal cells, we are telling, they are big. They are having prominent nucleus. They have abundant cytoplasm. They might have more than one nucleus, and they act as nutritive tissue for the inside sporogenous tissue. Now, if we have to see the tapetal cells inside, so we are telling these are the big tapetal cells which are helping in the nourishment of the sporogenous tissue. And we are telling it might have more than one nuclei also, and we are telling it is having abundant food reserves because it is a nutritive tissue. It should have abundant food reserves, abundant cytoplasm. They have more than one nuclei also. Now, these are the 4 layers which come under the anther wall. Epidermis, the outermost layer, it helps in protection. Endothelium, the second layer, hygroscopic cells, and they play important role in dehiscence. The third layers are called middle layers. 1 to 5 layers will be there, loosely packed parenchymatous cells. The innermost layer is the tapetal cells. They are thick, prominent nucleus, abundant cytoplasm, more than one nuclei also will be there. Now, think, how are they able to get more than one nuclei? Means in the embryonic stages, when the cells are fusing, when the tapetal cells are fusing, their cytoplasms will fuse, but the nuclei will not fuse. So, that is why after development, they will have their separate nuclei only. So, that is why it is having more than one nuclei. Inside this, what did we call this as? Microsporangium. Now, this microsporangium, what does it contain inside? The microsporangium, inside it contains sporogenous tissue. If it is a young anther, it contains sporogenous tissue. So, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 20, 24. So many. I mean to say that there are so many cells here. There are so many cells here. If it is a young anther, you call it as a sporogenous tissue. If it is a matured anther, then this sporogenous tissue converts into microspore mother cell or pollen mother cell. They undergo meiosis to make the pollen grains. Okay. So, inside this is the sporogenous tissue. The sporogenous tissue is nourished by these tapetal cells, and this sporogenous tissue, we always know that the male gamete production will be enormous because the male gamete has to travel and reach the female gamete. So, it is a hit or miss affair, right? So, that is why male gamete production will always be abundant, more in number. So, if you want those many male gametes, then each cell of a sporogenous tissue should be a potent spore mother cell. Each cell of the sporogenous tissue acts as a potent spore mother cell, and that spore mother cell undergoes meiosis. The spore mother cell undergoes meiosis to make pollen tetrad. Okay. So, understand this. Did you understand this? We talked about stamen. We talked about microsporangium also. Stamen has filament and anther. So, this is the filament. The distal end of the filament is attached to the thalamus or the petal or the tepal. The proximal end of the filament is attached to anther lobes. Usually 2 lobes, we told, bilobed nature. The bilobed nature will be very much clear when you make a transverse section. This is what is the transverse section of the anther. So, when you make a transverse section, you can see it is tetragonal, 4 sides are there. At each corner, it is having one microsporangia. So 2 lobes are there, so it's called dithecus. 4 sporangia are there, so it's called tetrasporangiate. So what word we can tell? Generally it is lobes, so it's called dithecus, having 1, 2, 3, 4. So it's called tetrasporangiate. Take a screenshot, then we will continue. Right, so can I erase this? We are telling that in most of the cases, the anther has 1 lobe and 2 lobes, and each lobe contains 2 microsporangia. That is a general condition. But science without exceptions is not possible. Everywhere there are exceptions. That means there are some stamens which have only 1 lobe. If 2 lobes are there, 4 microsporangias are there, then that is called as dithecus. Tetrasporangiate, we will write it here again. Yeah. So what is a normal condition? The normal condition is having 2 lobes and 4 microsporangiums. It is called dithecus tetrasporangiate. But there are some members which are belonging to the family Malvaceae. A prominent example is China rose and lady's finger. They have only 1 lobe, children. They don't have 2 lobes like that. The anther has only 1 lobe. If 1 lobe is there, monothecus. If only 1 lobe is there, 2 microsporangiums only will be there, then it is called as bisporangiate. So we can tell in Malvaceae. Example for Malvaceae is hibiscus. We call it as China rose. One more example is lady's finger. Now in this, what do we find is it's having only 1 lobe. Only 1 anther lobe is there. So then only 2 microsporangiums can be there. So 1 lobe is called as monothecus and bisporangiate. Monothecus and bisporangiate condition. Where do we find it in? China rose and lady's finger. Occasionally, if you see in Wolfia plant, 1 anther lobe will have only 1 microsporangium. So if we see in Wolfia, what is Wolfia? So Wolfia is the smallest angiosperm, right? So we can tell that Wolfia is the smallest angiosperm. Now what is the speciality of this Wolfia? In Wolfia, 2 lobes will be there. But each lobe has only 1 microsporangium. In Wolfia, each lobe has only 1 microsporangia or 1 microsporangium. Understood? So all these are the exceptions. So, but the general case is dithecus tetrasporangiate. Now I told a microsporangium contains sporogenous tissue. If the microsporangium is young, it contains sporogenous tissue. If it is matured, then it develops into pollen sac. So if it is young, the microsporangium, what does it contain? If the anther is young, then that circular microsporangium contains sporogenous tissue. It contains, in the previous slide we have seen that green colored cells are sporogenous tissue. Sporogenous tissue, sporogenous cells will be there. Whereas if it is a matured anther, then the microsporangium will contain pollen grains and that is called as pollen sac. Then we call the microsporangium as pollen sac. And what does it contains then? Pollen grains. It contains pollen grains. Understood? Now, if we have to talk about the structure of Yeah, we discussed about stamen. We discussed about microsporangium also. Microsporangium, it will be circular in outline. Microsporangium will be circular in outline, and this microsporangium is surrounded by 4 layers we discussed. The outer layer is epidermis, second layer is endothelium, third layer is middle layers, the fourth layer is tapetum. So we told in microsporangium, the outer 3 layers. What are the outer 3 layers? Epidermis, endothelium and middle layers. All the outer 3 layers help in dehiscence of the anther. All the outer 3 layers, what is their role? They help in dehiscence of anther. Whereas the inner layer, if you see, they are called tapetal cells. They contain more than 1 nuclei. Now, what is the role of the tapetum? So, tapetal cells, let us list down. It has many functions to do. Let us list down the functions of the tapetal cells. One function, we already know that the tapetal cells act as a nutritive tissue for the developing for the pollen mother cell and the pollen grain. That is one role. What can we write? So tapetal cells provide nourishment to whom? To the pollen mother cell and the pollen grain also. It provides nourishment to pollen mother cell and the pollen grain. This is one role of the tapetal cells. Again, the outer 3 walls are breaking, they are dehydrating and they are breaking. The tapetal cell also should dissolve. When the tapetal cell is getting dissolved, then it produces granules, lipid-rich granules, which are called Ubis bodies. These Ubis bodies, I told, they are rich in lipid, they are rich in carotenoids. Then they will make the outer coating of the pollen grain, which is called as exine. It makes that Sporophololine material. At the time of dehiscence, when the tapetal cells are degenerating, then they secrete, they produce lipid-rich Ubis granules. So tapetal cells produce lipid-rich Ubis granules. And I am telling that these Ubis granules, what do they make? They make the Sporophololine, which is the outer coat of the pollen grain. These lipid-rich Ubis granules, they make Sporophololine. What is the Sporophololine? It is the outer coat of pollen grain. Sporophololine, it makes the outer coat of pollen grain. So, the first function of tapetal cells is to provide nourishment to the spore mother cell and the pollen grain. The second function is to secrete Ubis granules, which will make the outer wall of the pollen grain. And entomophilous flowers, there the pollen grain should stick to the insect body so that it can carry it till the stigma. So then the sticky nature of the pollen grains is called as pollen kit. So these Ubis granules, they also help in making the pollen kit for insect pollinated flowers. So they make pollen kit. Where? In insect pollinated flowers. Pollen kit in insect pollinated flowers. This is one more role. Not only this, it secretes an enzyme called Calase enzyme. Now, if we see in NCERT, they have given a microspore tetrad here, and they showed this is one microspore, second microspore, third microspore and fourth microspore. All these 4 microspores are surrounded by a layer. This is a callose layer. Only when this callose layer dissolves, then the microspores can come out, right? So then this callose wall of the microspore tetrad is dissolved by the Calase enzyme secreted by the tapetal cells. So we can write, so the tapetal cells secrete. What do they secrete? They secrete Calase enzyme. They secrete Calase enzyme to dissolve the callose wall of the microspore tetrad. It secretes Calase enzyme to dissolve the callose wall of where? microspore tetrad. Children, what did we discuss in this board so far? We were talking about the normal condition of the anther lobes. 2 lobes means dithecous. Each lobe containing 2 microsporangia, 2 here means tetrasporangia. When we see the cross section, we have seen clearly the tetragonals, the microsporangias are at the corners and they are spherical, that we saw. So, we are calling it as dithecous tetrasporangiate condition. But in a family called Malvaceae, like China rose. And one more example, lady's finger. Only one anther is there, one lobe is there, one lobe, then only 2 microsporangia, one lobe, monothecous, 2 microsporangia, bisporangiate. So in Wolfia, there's another extreme. In Wolfia, there are 2 lobes, but each lobe will bear only 1 microsporangia. Wolfia, which is the smallest angiosperm, it has in each lobe, it has only 1 microsporangium. Now, young microsporangium, inside it contains sporogenous tissue. Matured microsporangium, it contains pollen sacs and pollen grains. The microsporangiums, all the outer 3 walls, they help in dehiscence, splitting of the anther walls to release the pollen grains. Tapetum has many roles. First function, provides nourishment. Second function, we are telling like it secretes Ubis granules which will make the Sporophololine, the outer layer of the pollen grain. Third one, it makes Pollen Kit which helps in insect pollination. Fourth one, it secretes a callus enzyme which dissolves this callus wall so that the microspores can be released out. Take a screenshot fast. Right. Now, so we talked about stamen, we talked about microsporangium. Now we will see that the microsporangium has sporogenous cells. These sporogenous cells will undergo microsporogenesis, and which is a meiotic division, so which produces 4 microspores. We'll talk about microsporogenesis now. So do you remember we told sexual reproduction involves 3 phases. Pre-reproductive phase. In pre-reproductive phase we told it involves gametogenesis and gamete transfer. Under gametogenesis we told microsporogenesis which happens in the stamen and microsporangium. Megasporogenesis which happens inside the ovule. Now let us talk about microsporogenesis. We discussed about the structure of microsporangium. Now let us talk about the process of microsporogenesis. What is microsporogenesis? It is the process of formation of microspores from microspore mother cell or pollen mother cell by doing meiotic division. So then microspores will be haploid. Okay, what is microsporogenesis? It is the process of formation of, I told, haploid microspores. It is the process of formation of haploid microspores from where? From diploid microspore mother cell or pollen mother cell. From diploid, we can write pollen mother cell, or you can call it as microspore mother cell. Now, from diploid you are getting haploid structure, means involving meiosis. Okay, diploid mother cell by meiotic division is called microsporogenesis. The process is called microsporogenesis. Now, this is the microsporangium. The inner content of the microsporangium, these are the sporogenous cells, sporogenous tissue. So in the previous thing we counted how many cells are there in the sporogenous tissue. So many will be there, right? Now we will tell, I told you that the male gametes should be numerous because they have to travel. So they might reach the stigma, they might not reach the stigma. So then if you need to produce many microspores, means each cell of the sporogenous tissue is potent pollen mother cell. So we will not waste single cell also. We will tell each cell of this sporogenous tissue. Each cell of the sporogenous tissue, it is a potent, it is a functional spore mother cell. It is a potent pollen mother cell. It is a pollen mother cell, and we told the ploidy is 2n, each cell is 2n. Then it undergoes meiotic division. Here is the cell, it has undergone meiotic division, then it made 1, 2, 3, 4 microspores. So one pollen mother cell after meiosis, it makes 4 microspores. This process is called microsporogenesis. It makes 4. What are these called? Microspores. They are haploid. This is diploid. So they'll ask you questions like, for formation of 100 pollen grains, how many microspore mother cells are required? For formation of 100 pollen grains, how many pollen mother cells are required? This is your question. You are going to post me the answer in the comment box. You're going to post me the answer in the comment box. So this is your clue. So I'll tell this in the next class. Like if you are not able to answer, we will talk about this in the next class. So it will make microspores. These microspores can be arranged in different patterns. Like the arrangement of those 4 microspores, it can be like this. So 1, 2, 3, 4. This arrangement is called as isobilateral arrangement of microspores. Another arrangement is also there. In that arrangement, so 1, 2, 3 cells are here. The 4th cell is behind it. This is called tetrahedral. Children, tetrahedral arrangement is a common type of microspore tetrad. This is a most common type of microspore tetrad arrangement. Linear tetrad is also there. So 1, 2, 3 and 4. What is this called? This is linear tetrad. T-shaped tetrad is also there. In the T-shaped tetrad, so 1, 2, 3 and 4. It looks like a T. What is this called? T-shaped tetrad. Now, another one is also there. We call it as dicuspate. In dicuspate tetrad, it will be like 1, 2, 3. The other one is behind it. So, these two at the right angles of that, you have the 4th one. So, what is this called? This dicuspate tetrad. So, from one pollen mother cell, when the microspores are formed, from one pollen mother cell, when microspores are formed, they can be arranged like this, isobilateral arrangement. They can be arranged like tetrahedral arrangement. This is a common type. They can be arranged in the form of linear also, T-shaped also, dicuspate also. So, all these are arrangements of microspore tetrads. Now, this is surrounded by a callose layer, right? We are telling that it is surrounded by callose layer. And then we told that the tapetal cells will secrete callase enzyme. They will secrete callase enzyme and they dissolve this callose layer. They dissolve this callose layer. When the callose layer gets dissolved, each microspore will be released out like this. So, what is this? This is the microspore, which is haploid. It's released out when the callase enzyme dissolves this callose wall. Now, this microspore develops into pollen grain. It develops into pollen grain. So, about pollen grain, we will discuss in the next lecture. Now, what did we discuss in this lecture? We discussed about stamen having 2 parts, filament, sterile part, anther lobes, fertile part. Anther lobes contain 2 lobes. In the lobes, microsporangiums will be there. The wall of the microsporangium is epidermis, endothelium, middle layers and tapetum. Tapetum functions we have seen, and then the young anther, young microsporangium contains sporogenous tissue which is diploid. It undergoes microsporogenesis to make microspores by doing meiotic division. So, one pollen mother cell will undergo meiotic division to make 1, 2, 3, 4 microspores. Now, we are telling that since microspores will make pollen grains should be numerous in number. So, each sporogenous cell is a potent, microspore mother cell. So, hope you like the lecture. The content is informative, up to the mark of the need. So, any suggestions, kindly post them in the comment box. So, if you like the content, like, share and subscribe to my channel. Thank you.